Good morning. Welcome to our last class of the semester officially. I'll be on here again Monday night at our usual time, 7:30 to answer any last minute final exam questions or that kind of stuff. But this is our last session of new information. So getting close to the end there. Hope everybody's doing okay. Today we're going to be talking about molecular genetics, aka nucleic acids, DNA and RNA. All right, so let's get started. So I have a rough outline of some notes here. Again, just like with the proteins, you don't have to complete this and turn it in. There's no assignment for this, but it is posted on Blackboard if you want to follow along. So with nucleic acids, we're looking at a similar kind of a system as with the other biological molecules we've looked at so far. So we've got a small building block of some sort that then gets expanded into this larger molecule. So in this case, our smallest little building block pieces are called nucleic acids, nucleic acids or nucleotides. I think nucleotide is the better term. Nucleic acid is usually the whole thing together. So nucleotides are these things. And we're going to go through their structure. I'll zoom in and kind of show you a little more detail of what that involves. But let's jump ahead a little bit and just look at where we're going first. So you've probably seen these kinds of shapes before: DNA and RNA, deoxyribonucleic acid and ribonucleic acid. And you may know something about how they function in cells. We're not really going to focus on the function part in this class because it's chemistry. But if you go on to take biology or biochemistry, microbiology especially, you'll learn that in great detail. But DNA contains the genetic information of the cell, and RNA, there's a few different types of RNA that are involved in cells, but the DNA is in the nucleus, and the RNA is outside the nucleus. There's ribosomal RNA, and then there's RNA in the cytoplasm and stuff, and it's used for transferring, it's used for cell division, but we're going to kind of zoom in and focus on the chemistry right now so you know what these things are actually made of. All right, so as this document says, there are basically three parts of a nucleic acid. You've got the nitrogen-containing bases, and we'll look at those structures in a second. So there, you probably can't see this. Let me get this a little bit. What am I? Sorry, trying to get this thing show up a little bit better. All right, there it is. So this is from the book that's linked on Blackboard. So here are the three major parts of the nucleotide. In this case, this is the DNA nucleotide. So you have a phosphate. So that's phosphorus with some oxygens, and that's connected through a ester type linkage. So the way we talked about ester functional groups, carbon double bond to oxygen, and then another oxygen coming off that carbon. Well, this is a phosphate or phosphonate ester that is connected in a similar way. So then you've got some carbon over here, and then we have our sugar, our carbohydrate. In this case, this is deoxyribose, which is a pentose sugar, of a 5-carbon sugar, 1, 2, 3, 4, 5. And then that is connected to what we call the base, which is this molecule that has a bunch of nitrogens in it, and it acts as a base in terms of like acid-base properties. So we usually call those the bases. And there's a few different types of these. So the sugar itself, this pentose sugar, is called deoxyribose. There's also just ribose without the deoxy, where this lower, I don't know if you can see that. Yeah, this lower H is replaced with an OH. So in the original sugar, we might call it plain old ribose, you have an OH here. In deoxyribose, the oxygen's gone. That's the deoxy, and you have a H instead. We should also note that in this diagram, these extra sticks here are not carbon. So this is not a traditional line structure in that way. These are actually hydrogens. So it'll be hydrogen here, 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 and here. So there's our sugar. And then we've got the base. So let's take a look at some of the different bases. So in DNA, there are 4 different bases that you can have connected to your ribose sugar. Cytosine, thymine, adenine, and guanine. And 2 of them are the pyrimidines, which are just means they're these 6-membered rings that are with 2 nitrogens in them. And then you have the purines, which are these bigger ones. So cytosine, thymine, adenine, and guanine, and those are generally abbreviated by their first letters, C, T, A, and G. In RNA, instead of thymine, that's why it says in DNA, instead of thymine, we have a similar base called uracil with a U. All right. So then how are these things put together? All right, so remember this piece here, which is composed of phosphate, sugar, and base. That's our nucleotide. And that links together with another one to form what we call a phosphodiester. So let's look at the highlighted atoms here, and you should see something pretty familiar. We have an OH from the phosphorus here, from the phosphate here, and we have the H from the OH on the sugar, and those are combining to make water. And then the resulting, so there's our water, and then the resulting atoms are connected together. So we've made an ester linkage, and this is the same kind of a dehydration reaction that we've seen now multiple times in different scenarios, particularly with sugars, but also with lipids. So we've made another ester. It's a diester because we've got an ester going this way to this as part of that original nucleotide, and then we have our new ester that links the 2 nucleotides together. So that's kind of a special thing about phosphorus's ability to bond that carbon wouldn't do in exactly the same way. So having the phosphate here allows us to have a diester, 2 different linkages. And then much like our other polymers, like the amino acids that we saw last week or Monday, we still have an open spot here and an open spot here for more things to connect. So they haven't stopped there. We're not just stuck with the reaction between these 2 nucleotides. We can keep adding more nucleotides to either side. And this is how one strand of DNA would be constructed. So this would happen over and over again. And depending on the order of the bases, that gives us what we might call our genetic sequence, our sequence of nucleotides, A's and T's or G's and C's that form the entire strand. All right. So that's the chemistry of how these things are put together with that dehydration reaction. And then I think I'm going to move back over to the book because I like the pictures a little bit better there. Okay, so now we've made a strand doing these reactions. Here's another example of that. So you have your phosphodiester connecting your sugars, and then the bases are connected to those sugars. And these bonds are drawn in a kind of exaggerated long way so that we can really see see everything. But normally it would be, normal bond lengths. Okay. And so now here comes the double helix, and there's some interesting history here if you want to look at how that was discovered. But let's just kind of zoom in and take a look at what that double helix really looks like. So we've all seen, probably this kind of a picture where you've got your, your spirally looking thing with the sticks in the middle. That's kind of like the classic picture of DNA. So let's zoom in on that a little bit. DNA is actually, or the 2 strands are directional and opposite. So on one side you have you can think of it as going phosphate, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And then the other side, it's going sugar, phosphate, sugar, phosphate, sugar, phosphate. And because of how that's connected to the sugar, those are actually different. So they're like flipped over. Let me see if I can make this bigger. I can't really make the picture bigger, but hopefully that's, yeah, I think you can probably see that. So we've got, on the left side, we're starting at the 5 position here, which is the carbon coming off of the ring. And on the other side, we're starting at the 3 position, which is right over here, where the OH comes directly off the ring. So there's a carbon, and there's the O. And so those are different sides. So one strand's going one direction and one's going the other direction. And that allows the bases to line up in this particular way. And you can probably guess what these dotted lines refer to that hold this thing together. If you look carefully at those atoms, we've got an NH and an O, NH and an N, NHO, NHN, and those are hydrogen bonds. So those are the intermolecular forces that we've been talking about quite a bit for a while, known as hydrogen bonding. And those are the forces that holds the 2 strands of DNA together. Remember, hydrogen bonds are pretty strong intermolecular interactions, but they're not as strong as covalent bonds. So those interactions are not as strong as the solid bonds that we see in the rest of the structure, but they're still able to hold these things together. And what you can see here is that certain base pairs always line up together. So adenine and thymine are lined up just right. And then cytosine and guanine are also lined up just right. And if this were RNA, then, and I think I have a picture of that over here, then instead of thymine, we'd have uracil, which is this. So very similar, just with an H instead of a CH3 there. And that one would line up with adenine instead. So here's our DNA. And so in replication, what happens is the DNA strands break apart along those hydrogen bonds, and the RNA comes in and matches up, or is made to match up, and that's a way of transcribing. That's why it's called transcription. So you're taking the original information from the DNA, you're making some RNA that matches it basically, and then the RNA is coming apart and making the new strand somewhere else. Okay, so that's our nucleic acid strand. The whole thing is the individual strands, which are made of a phosphate or sugar phosphate backbone, which are alternating phosphates and sugars connected by a phosphodiester. Then those are connected with the bases as part of those nucleotides, and the bases attract each other, hold each other via these hydrogen bonding interactions. And there's a close-up again of that hydrogen bonding interaction and how that thing works out. All right. So that's what you need to know about nucleic acids. Again, as usual, check on Blackboard for the specifics of the outcomes that you're supposed to. Hold on. Wrong thing here. Oh, you know what? I don't know if I'm going to be able to get it to show up here. Let's see. There it is. Okay. So make sure that you understand the difference between DNA and RNA, which is mainly where they are in the cell, what they're for, and structurally the importance is the difference in the sugars, deoxyribose versus ribose, and the difference in the bases, thymine versus uracil. And then you should be able to identify the structure, where are the hydrogen bonds, and how is the structure built, just what we've talked about with the phosphodiesters, and how those nucleotides come together to form the full structures. Okay, so that's it for nucleic acids. If there are any questions, go ahead and post them or send them on Discord. So today we will be in doing review on campus for those of you who signed up. There is still space, so if you want to just show up, there should be room unless everybody shows up at noon. I think we'll be okay. If you want your exams back, if you haven't gotten those back yet, I still have some exams, exam 2s, so you can grab those as well. And yeah, if you feel like you're good to go, you don't need to come to campus today. We'll meet back here on Monday evening at 7:30 for any last minute questions, and I'll see you at the exam, which will be at our normal Wednesday noon time next week. So, all right, see some of you today and the rest of you next week. Bye.